Hello everyone, today we'll be looking at the influence of submarine canyons on zooplankton communities in the east coast of South Africa. So what are submarine canyons? These are steep valleys with a V-shaped cross-section that occur on continental margins. They are important conduits of sediment from the continental shelf towards the deep sea. They can be connected to fluvial systems, but most of them in the northern KZN are not. So we have two types of submarine canyons. We have mature shelf breaching canyons, which goes all the way to the shelf, and we have blind canyons, which only occur in the slope breathing. The formation of submarine canyons can be linked to mass erosions that occur in the last ice age. They can form to fluid seepage in the slope region, which causes a submarine landslides. They can also be caused by turbidity currents or tectonic activities such as earthquakes. Zooplankton are weakly swimming animals that are found floating in a variety of aquatic environments. They play an important role as primary consumers of phytoplankton. Thus, they maintain the marine food web. To understand the effect of submarine canyons on zooplankton communities, we have to look at their flow dynamics. Submarine canyons disturb the path of the current flowing along the coast. This can result to canyon-induced upwelling, which can result to increased primary productivity. Also, this upwelling current can transport slow-swimming zooplankton from the depths towards the canyon head. Also, the cyclonic flows and downwelling can act as trap of zooplankton and then makes it available for animals in the surface. Some of the threats to submarine canyon ecosystem include destructive fishing methods such as trawling. So from this map you can see that submarine canyons are targets to fishing. With the sediment that they trap, they also collect plastic pollutants and chemical pollutants as they occur very close to the coast. So in this project, we wanted to test the canyon effect hypothesis. So we wanted to see if submarine canyons support higher abundance of zooplankton. Also, we look at the biomass and the biovolume in the canyon heads. So we want to see also if the submarine canyons support different communities compared to their adjacent shelf area. And we will also look at what are the environmental or abiotic factors that control the observed patterns in zooplankton communities. The study area is in the northern KZN. It is characterized by a very narrow continental shelf, and it has the warm and the canyons they are washed by the warm Agalas current that flows across them. Here we have a sampling design in right canyon. So on each canyon head, we sampled 3 stations and sampled 3 in the shelf region. The samples were collected on Angra Pequena during the ASEP Canyons Connection cruise in June 2018, and we also collected sample in June 2019. We used a bongo net with 200 micron mesh to collect zooplankton, and the net is equipped with a flow meter to measure the water sampled. And at each station we also collected environmental variables using a CTD, and the CTD has Niskin bottles that are used to collect water sampling. After the samples are collected, they are preserved with a 4% formaldehyde for further processing in the lab. And in the lab, we cycle the samples in seawater in a measuring cylinder to measure the zooplankton cycled volume, and then we divide that by the volume filtered to get the bio volume. And we also dry the samples and divide the dried weight by the volume filtered to get the biomass. Abundance and diversity analysis involves a lot of counting and identification of zooplankton, and abundance is number of individuals divided by the volume filtered by the net. So we analyzed the biomass, biovolume, and diversity indices on SPSS for univariate analysis, and the species abundance was analyzed on primer. From this temperature depth profile, we can see that submarine canyons were a bit cooler compared to their adjacent shelf area, and this could be an indicator of a cold deep water from the canyon depth towards the canyon head. And at an average of 0 to 40 meter depth, we can see that the deep heart was the warmest of these canyons, and Leven was the coolest, and somewhere in the middle was the right canyon. And we can see that there was a significant canyon effect at deep heart and right canyon. More oxygen were observed in canyons that had cooler water. The oxygen differed significantly between the stations, but there was no significant canyon effect. In terms of salinity, a deep heart, which was the warm station in this case, had the lowest salinity, and there was a significant canyon effect only at deep heart. When we looked at the chlorophyll A profile, you can see that the canyons, especially at Leven, had higher chlorophyll A, indicating higher primary productivity of the canyons, and it was significant between the stations and between the canyon and non-canyon. A similar trend was observed with biomass. So we found a significant difference between the stations, and there was a significant canyon effect in right and 11 canyon only. And in Tibet, we didn't find any significant difference in biomass between the canyon and canyon station. So the biomass and the biovolume abundance can be linked to the higher chlorophyll A observed in this station. Abundance showed a different trend from the biomass and biovolume. We didn't find any significant canyon effect, and I think the reason for the mismatch in the abundance and biovolume and biomass is because of a high occurrence of small individual but in very large numbers in the non-canyon stations. Some of the abundant coppepods that we found include Paracalanus, which can be identified by that very small leg there. That's Acrocalanus, and this is a type of eucalyptus, and this is Onkea. Okay, I wish I would share my whole gallery with you guys, but the time doesn't allow. But some of the rare species that I found is this type of Oitoina. I think I found one or two like this, and this is a Harpacticoid copperpot. It's Meracia species, and this is Pseudodeltomas species. Some conclusions that can be drawn from this is that environmental variables do differ between the canyon heads and the adjacent shelf area, and the submarine canyons do influence the quantity in terms of biomass and biovolume of zooplankton around them. And thank you for riding this submarine roller coaster with me. I am ready to take your questions.